In 1994, 224,000 Americans got violently sick from a single food source.
Right.
And I mean, a massive foodborne illness is always alarming. But here is the staggering historical fact hidden in the records of this specific outbreak.
Right.
The food they ate was ice cream. And that ice cream had already been pasteurized.
Yeah, that's the crazy part.
Right. The manufacturer had executed a deliberate, scientifically proven thermal kill step. I mean, it was designed to just obliterate any pathogens. This organism faced a direct assault meant to destroy it, totally bypassed it, hitched a ride on a massive commercial scale, and triggered one of the largest foodborne outbreaks in United States history.
It is a terrifying testament to just how resilient and opportunistic this organism really is. Like, we build these massive, impenetrable industrial walls to keep it out of our food supply, and it constantly finds the microscopic cracks.
It really does. So welcome to the deep dive. The mission today is to completely remap your understanding of an organism you probably just associate with, a bad batch of potato salad at a summer picnic.
Yeah, that reputation is a massive understatement.
Oh, totally. We are tracking a single bacterial species that can cause a miserable weekend of stomach cramps, sure, but also the life-threatening, history-altering disease known as typhoid fever.
Oh.
Okay, let's unpack this. What exactly are we dealing with when we say Salmonella?
Well, at a fundamental level, Salmonella is a genus of rod-shaped gram-negative bacteria.
I always hear that term gram-negative thrown around in medical context. What does that actually mean for us?
It means the bacterium essentially wears a double layer of armor. Gram-negative bacteria have an inner cell membrane and an outer cell membrane, which makes them inherently much harder for antibiotics and our own immune systems to break open.
Oh, wow.
Yeah. And additionally, they are facultatively anaerobic. In microbiology, that means while they can use oxygen to survive, they absolutely do not need it.
Which I imagine is a pretty handy trait if you are trying to survive deep inside the human gastrointestinal tract.
Precisely. It thrives in that oxygen-deprived environment. It belongs to the family Enterobacteriaceae, meaning it's a genetic cousin to E. coli. But within the Salmonella genus, there are really only two formally recognized species. You have Salmonella bongori and Salmonella enterica. Almost every infection you have ever heard of is caused by just one of those, and that's Salmonella enterica.
Which brings us to a frankly bizarre origin story. Because the very name Salmonella is essentially a case of stolen scientific identity.
It really is.
Right. So in 1885 at the USDA, there's this brilliant young researcher named Theobald Smith, and he was investigating an outbreak of hog cholera. He did the grueling laboratory work to actually isolate the bacterium from sick pigs. But his supervisor...
A veterinary pathologist named Daniel Elmer Salmon.
Exactly. Salmon published the findings under his own name.
And because of the conventions of the time where the lab director often just took top billing, the organism was named Salmonella choleraceus in Salmon's honor.
It's so unfair.
Yeah, Smith did all the heavy lifting, but Salmon got immortalized in medical textbooks. Though, to be fair, Theobald Smith later co-discovered that ticks transmit Texas cattle fever, so his legacy did survive. He was a genius in his own right.
Still, the naming injustice really stands out. But the timeline gets even weirder, because the strain that causes typhoid fever was already known before Smith even looked at a pig, wasn't it?
It was, yeah. During the germ theory revolution of the 1880s, a German pathologist named Karl Joseph Eberth identified the typhoid-causing organism in the spleens and lymph nodes of victims. That was in 1880. And by 1884, George Gaffke had successfully cultured it.
So what do they call it then?
At the time, it was classified as Eberthella typhi. So the person who discovered the cause of typhoid and the namesake of the genus it eventually ended up in are two entirely different people, and neither of them discovered the first Salmonella strain.
It's just a taxonomic mess.
Yeah.
And if you look at older medical literature, or even some current public health warnings, they talk about Salmonella as if it's dozens of different species. You'll see Salmonella typhi, Salmonella typhimurium. Why is the naming so contradictory?
It basically comes down to a system developed in the 1930s and 40s called the Kaufman-White scheme. Scientists back then didn't have DNA sequencing, obviously, so they classified these strains based on their physical surface antigens.
And an antigen is?
It's basically a protein flag on the outside of the cell that our immune system recognizes. They looked at the O antigens, which are on the main body or cell wall. the H antigens, which are on the flagella.
The little tails the bacteria use to swim, right?
Exactly. And for typhoid, the vi-capsular antigen, which is a special protective shield. Because these strains had different flags and caused different symptoms, microbiologists treated them like entirely separate species.
But then whole genome sequencing arrives in the 2000s, and we realize we've been looking at this completely wrong.
Right. The genetics revealed that most of these so-called separate species were practically identical at the DNA level. They are just slight variations on a single organism, Salmonella enterica. So technically, the correct, scientifically accurate name for the bacteria causing typhoid fever is Salmonella enterica subspecies Enterica serovar typhi.
Yeah, nobody is saying that in an emergency room.
Definitely not. What's fascinating here is that clinically, we still use the old shorthand. We just say Salmonella typhi because it's more intuitive for doctors and patients. But this creates a real ongoing headache in the scientific literature because you have the exact same organism referred to by wildly different names depending on the decade the paper was published.
Well, the taxonomic trickery is nothing compared to how this bacterium tricks the human body. Let's walk through the actual mechanism of invasion. Say you accidentally ingest a contaminated piece of food. Step 1, the bacteria hit the stomach, which is literally a vat of hydrochloric acid designed to melt down things exactly like this.
And Salmonella anticipates that. It triggers an acid tolerance response. The bacterium fundamentally alters its own gene expression, producing specialized proteins that actually pump acid out of its own cell.
Wow.
Yeah, it does this to survive that shockingly low pH environment just long enough to pass through the stomach and reach its true target. That's the small intestine, specifically the lower section called the ileum.
And once it gets to the ileum, it has to, break into human tissue.
Right. It seeks out specialized epithelial cells called M cells, which sit over your Peyer's patches. Those are the lymphoid tissue in your gut wall. To get inside, the bacterium uses a microscopic structure called a type 3 secretion system, or T3SS1. This is encoded on a cluster of genes called Salmonella pathogenicity island 1.
Okay, I am trying to visualize this. If the bacterium is a burglar trying to get into a house, instead of picking the lock, it slides something under the door to drug the homeowner, and the homeowner just opens it up.
You're on the right track, but it's much more forceful. The type 3 secretion system is essentially a molecular syringe. The bacterium docks with the outside of your cell and injects its own effector proteins like SIPA and SOPB directly through the keyhole. These proteins chemically hijack the host cell's actin cytoskeleton.
The cytoskeleton, that's the internal scaffolding that gives a human cell its structure, right?
Exactly. The injected proteins force that scaffolding to rapidly collapse and rebuild itself. So the surface of the human cell physically bubbles up and folds over. It's a process we call membrane ruffling. The human cell reaches out and aggressively engulfs the bacterium, pulling it inside.
That is wild.
Yeah, it's known as the trigger mechanism of invasion. The bacterium forces a cell that isn't supposed to eat things to swallow it whole.
Okay, so the burglar's inside the house. What does it actually do next? Does it just start replicating and tearing the cell apart?
Not yet. Once inside, the bacterium is enclosed in a tiny bubble called the Salmonella-containing vacuole, or SCV. You would think it's trapped in a cage, but a second cluster of genes activates, SPI2. This genetic toolkit remodels the bubble into a highly fortified safe house. It allows the bacterium to survive and even replicate inside macrophages.
Wait, macrophages are the immune system's heavy hitters, aren't they? Their entire job is to consume and digest invaders.
They are, and Salmonella turns them into a high-security hotel. It's basically building a panic room inside the police station. From this point, the disease splits into two entirely different paths depending on the specific strain or serovar you've been infected with.
So what happens if it's the standard food poisoning strain?
That's path A. The non-typhoidal serovars like Typhimurium or Enteritis. This is the noisy path. Those effector proteins we mentioned earlier provoke your intestinal cells to release a chemical distress signal called interleukin-8. This acts like a flare gun, recruiting a massive flood of immune cells called neutrophils straight into your gut lining.
So it's just an all-out, localized war zone. And that massive immune response is what actually causes the terrible cramping, the fever, the bloody diarrhea.
Yes, exactly. The symptoms are largely your own body's scorched earth defense. But because it is so loud and contained to the gut, the body usually clears it in a few days.
Okay, but if you ingest a typhoidal strain, does that mean the gut warfare is just 10 times worse?
That is a very common misconception. No, path B, which is taken by serovars like typhi and paratyphi, is the stealth route. Early on in a typhoid infection, you often don't see severe diarrhea. It's characterized by a sustained fever, lethargy, and frequently constipation.
Wait, really? Constipation?
Yeah, because the bacteria aren't staying in the gut to fight. They stay quiet. They hitch a ride inside those macrophages we talked about, traveling through the lymphatic system directly into your bloodstream. And from there, they seed themselves in your liver, your spleen, your bone marrow, and your gallbladder.
But if they are floating through the bloodstream, how do they avoid getting ripped apart by the blood's natural defenses?
They deploy that vicapsular antigen we talked about earlier. This shield allows them to evade something called complement-mediated killing. The complement system is a network of proteins in your blood that normally act like microscopic landmines. They snap together to punch holes in the walls of invading bacteria.
Oh, I see.
Right. But the Vi shield physically blocks those proteins from attaching. This allows typhi to cause a prolonged systemic infection. And critically, unlike the non-typhoidal strains, which can happily live in chickens or pigs, typhi and paratyphi are entirely human-restricted.
Meaning they don't infect animals at all.
Correct. They have no animal reservoir. They spread exclusively through human fecal contamination.
Wait, if they're specifically setting up camp in the gallbladder, doesn't that explain Mary Mallon? Was her gallbladder essentially the home base for that massive outbreak in the 1900s?
It was. Mary Mallon, Typhoid Mary, was an Irish immigrant cook working in New York, and she was the textbook definition of an asymptomatic carrier. She harbored Salmonella typhi in a biofilm directly on her gallbladder. She felt completely fine, showed zero symptoms, but she was constantly shedding the bacteria into the food she prepared.
That's horrifying.
A sanitary engineer named George Soper eventually traced a cluster of typhoid cases back to her various kitchens. She was officially linked to at least 51 cases and 3 deaths, and was forcibly quarantined on North Brother Island for decades.
I mean, if we connect this to the bigger picture, Mary Mallon was one person armed with a wooden spoon in a single kitchen. But when you look at modern industrial vulnerabilities, the scale of the threat changes exponentially. Which brings us back to that 1994 Schwann's ice cream outbreak we started with. If they pasteurized the ice cream mixture, how did 250,000 people get infected with Salmonella enteritis?
Because the vulnerability wasn't the cooking process, it was what happened afterward. Investigators traced the outbreak to the massive tanker trucks used to transport the pasteurized ice cream premix to the freezing plants.
Oh no, let me guess.
Yep. Those exact same trucks had previously hauled raw, unpasteurized liquid eggs, and they hadn't been adequately cleaned in between shipments. The pasteurization worked flawlessly, but downstream cross-contamination reintroduced the bacteria to a product that was already deemed safe.
Wow.
Post-cooking vulnerability is the real Achilles heel of modern large-scale food systems.
And sometimes the threat isn't just an accidental oversight in cleaning. In 2008 and 2009, the Peanut Corporation of America caused a massive Salmonella typhimurium outbreak. It sickened around 700 people and was linked to 9 deaths. But the truly chilling detail from the internal records was that executives were shipping peanut paste that they knew had tested positive for Salmonella. They actively hid the lab results.
Yeah, that was a watershed moment in food safety law. The company owner, Stuart Parnell, received a 28-year prison sentence. It signaled that distributing known contaminated food wasn't just a regulatory misstep to be punished with a fine. It was a severe criminal act.
I want to spend a minute dissecting some common myths, because this organism is surrounded by them. For example, I've always assumed the only way I'm getting Salmonella is from eating raw eggs or undercooked chicken.
That's a huge myth. Historically, poultry and eggs are major vectors, sure, but they are far from the only ones. We have seen major outbreaks traced back to cantaloupe, cumin, pistachios, and even peanut butter. In fact, in 1975, the CDC had to ban the commercial sale of small pet turtles because children were putting them in their mouths and triggering waves of pediatric salmonellosis.
Yikes. Well, as long as I cook my chicken to 165 degrees, I am bulletproof, right?
Only if you don't ruin your own kill step. If you take that perfectly cooked chicken and put it right back onto the unwashed cutting board you used to prep the raw meat, or if you touch a salad with contaminated hands, the heat of the oven doesn't matter anymore. Just like the Schwann's ice cream trucks, you've cross-contaminated the final product.
Here's where it gets really interesting. So if I end up with the noisy food poisoning version of this, I'm immediately going to urgent care for antibiotics. Just carpet bomb the gut and get it over with.
You'd think that would be the best move, but most modern medical guidelines actually advise strongly against antibiotics for a routine, uncomplicated case in an otherwise healthy person.
Wait, really? Why wouldn't I want to kill the bacteria?
Because the data shows it doesn't meaningfully shorten your symptoms. Worse, Taking antibiotics wipes out your native gut microbiome. Those are the good bacteria that are naturally competing with the invader for space and nutrients.
Oh, I see.
Right, so by killing off your own defenders, you give the salmonella free rein. You actually prolong the amount of time you are shedding the bacteria in your stool, keeping you contagious longer. And crucially, unnecessary antibiotic use directly fuels antimicrobial resistance.
And how exactly does that resistance happen? Does the bacteria just get stronger over time?
It's more insidious than that. Bacteria can engage in horizontal gene transfer. They contain small, circular pieces of DNA called plasmids that operate entirely separate from their main chromosome. If one bacterium figures out how to survive an antibiotic, it can copy that plasmid and simply hand the instruction manual to a neighboring bacterium.
So they just share the resistance.
Exactly. Back in the 1990s, we saw the spread of a strain called Salmonella typhimurium DT104 in cattle and poultry. It was nicknamed the ACSUT strain because it carried plasmids, making it resistant to 5 entirely different classes of antibiotics at once.
5 classes. So we're running out of weapons.
We really are. In 2016, there was a massive outbreak in Sindh province, Pakistan, of extensively drug-resistant, or XDR, typhoid. This strain had acquired resistance plasmids to almost every oral antibiotic option we have. A disease that used to be treated with a simple course of pills suddenly forced doctors to rely on intravenous carbapenems. Those are heavy-duty hospital drugs of last resort.
Yeah.
And this raises an important question. If we view this bug strictly through a Western lens as just a bad potato salad issue, we are completely blinding ourselves to the global stakes.
Because the burden looks completely different in other parts of the world.
Absolutely. In wealthy countries, non-typhoidal strains drive massive product recalls and a few days of misery. But in sub-Saharan Africa, there is a distinct and severe syndrome called invasive non-typhoidal salmonella, or INTS. Yes, in populations dealing with malnutrition, malaria, or HIV co-infection, these ordinarily mild strains don't stay in the gut. They invade the bloodstream directly. The mortality rate for INTS can run around 20%. It is a devastating hidden killer that looks absolutely nothing like our Western stereotype of food poisoning.
It really highlights how incredibly reactive our systems can be rather than proactive. Like, look at the regulatory failures here in the U.S. In 2010, there was a shell egg recall involving 500 million eggs from Iowa farms linked to a nationwide outbreak. That single event was the direct impetus for the FDA to finally mandate the egg safety rule for large producers. We literally waited for 500 million potentially infected eggs to hit the market before mandating a prevention plan.
Yeah, regulation acting retroactively, trying to close the barn door long after the bacteria has already escaped.
So what does this all mean? We started with a young researcher robbed of his scientific legacy, and we've mapped an organism that acts as a master of molecular disguise. It survives a vat of acid, injects serums through the keyhole to force our cells to swallow it, builds panic rooms inside our immune system, and splits its attack between explosive local warfare and stealthy systemic invasion. How do we even fight something that adaptable?
By tracking its genetics closer than ever before and understanding how it evolves. For instance, Shigella, a genomic neighbor in the same bacterial family, shows a textbook case of convergent evolution. It independently evolved a type 3 secretion system strikingly similar to Salmonella's. They are arriving at the same deadly evolutionary ideas.
Right.
To fight back, modern surveillance technologies like Genome Tracker and PulseNet are replacing those old serotyping tests to catch outbreaks at the DNA level before they spread. And globally, Gavi-supported typhoid conjugate vaccines, TCVs, are being aggressively rolled out in high-burden countries to combat those XDR strains.
It really is an invisible arms race. And as we wrap up today's deep dive, I want to leave you with a thought to mull over next time you're prepping dinner. We know that bacteria like Salmonella and its neighbor Shigella evolve these incredibly complex microscopic weapons, like the pathogenicity islands that build those molecular syringes. And we live in a world with a highly globalized food supply chain, where a single processed snack might contain ingredients sourced from 30 different countries. When you mix that level of global physical cross-contamination with bacteria that constantly swap and evolve genetic blueprints via horizontal gene transfer, well, what happens when a relatively benign gut bug floating around today stumbles upon one of these weaponized genetic blueprints tomorrow? It makes you realize that the constantly evolving arms race isn't just happening in a lab in Pakistan or an egg farm in Iowa. It's happening right now on your very own kitchen counter.